Hello, and thanks for joining us today. I'm Fenella Saunders. I'm the managing editor for American Scientist magazine, and I'm the vice president of the RTP chapter of Sigma Xi. Today we're really pleased to be talking to Dr. Yi Liu, who is a professor at the University of Illinois, and he's one of Sigma Xi's distinguished lecturers for 2013. He's going to be talking to us about his research, which is at the interface of chemistry and biology, specifically about his work on DNA nanotechnology.
Thank you for inviting me. It's a great honor for me to be here.
Most people don't think of sensors when they hear the word DNA. Can you describe how you started using them for that purpose?
When we started, we also was not, that's not our initial goal either. The reason we got into this field about using DNA as a sensor was actually, it was discovered in 1994 that DNA can not only be a regular genetic storage material like we all know, like a gene. The DNA can also be so-called metalloenzyme. They can bind the metal ions and using metal to catalyze reactions. So that immediately drew my attention because as a bioinorganic chemist, we're interested in metal side in biological systems, where the other part of my group interested in metalloproteins. It's exciting to me because now DNA can be like protein. It can bind metal ion to catalyze reactions. So immediately the question is that how many metal ions does it bind? What kind of metal ion it can do? Can we find the DNA sequences that bind calcium, bind magnesium, bind iron, bind lead, bind many other different metal ions? Can we select specific sequences for specific metal ions? So that was a completely fundamental research. And then it dawned to us, and particularly me in the conference, I realized that now I have a collection of different DNA sequences for different metal ions. Can we actually utilize not only for fundamental research to understand what's the sequence requirement, what's the structure for metal ion, but can also use it for detecting or sensing for different metal ions. Obviously, there's a wide variety of reasons for it because there's a nutrition, right? We also know calcium and iron is important. We want to know how they works, but more importantly, there are a lot of toxic metal ions, heavy metal ions, for example, lead and mercury. So that's why I think can we actually turn these fundamental research into something more applied that people actually can use for that.
Can you describe how your work crosses boundaries between biology and chemistry?
Now we can actually utilizing sensors to start really asking fundamental questions, right? Why iron is so good, why copper, and why all the calcium and magnesium, why they're performing their functions. We can using the sensor to detect in the cells, but now also can into the animals and also in the near future in the human beings. But more importantly, why toxic metal ions exert their effects. For example, why lead is so toxic. A lot of hypotheses have been made. Lead it does toxicity, for example, in the brains or in other places. But right now, there are very little information, if any, actually, about where does the lead go. We know have a whole blood. We can actually detect the whole blood in the urine, in the blood. But unfortunately, we don't have any information about the location. How does it change the function of these toxic metal ions, including not only lead but also mercury and cadmium, many other species. So now we actually can utilize what we have. The tools, basically chemistry tools, that we can make a sensor. Now we can actually go back into biology to really ask a lot of questions and getting a lot of information from biological systems.
Are there overarching principles to your research, specifically related to reversible dynamic building processes?
So far, anyway, is majority of the research has been focusing on how we can put everything together and a certain condition and form in certain beautiful nanoscale assembly. But the very few time, very limited research has been going on in terms of reversible assembly. How do you actually put the part, not only assemble it, but also take part away and put it back in the reversible way. But if you look at the biological system, it does it all the time. In transcription, translations, there's one factor, one component goes in, it goes and does its job, and then they finish the work, it go back, and then the second component comes in, and they're very elaborate, very kind of a controlled ways. So now can we do that? So that's what the research we have just published earlier this year. We can actually allow us to inspired by biology and try to learn actually how to do that. In fact, we use one of the fundamental part of the biological system, the biotin-strap-avidin interactions. We're able to use two different kind of either one's biotin, one's the thiobiotin, have a different, slightly different affinities. We can actually use that to be able to take advantage of that biological system, allow us to do some kind of nanoscale assembly in a reversible way.
How does one make DNA into functional DNA?
Right. So this is actually a very interesting question because so far anyway, it's relatively difficult to design it, quote unquote, by knowledge of computers. Although we're getting there. So what we actually do in the lab is actually using these combinatorial selections. We ask a very simple question: out of a large number of DNA library that consists of 100 trillion different sequences and then can we actually select out from that 100 trillion different kind of a library a small subset of sequences that are capable of performing certain functions, function like catalytic or function like binding, like antibody functions. So basically we can actually select out a different subset of sequences that are capable of performing the function. We do not actually know what sequence they are, but then we can actually obtain sequences, and that's what we want to do. And then now we can actually start analyzing, this sequences works, why it works. So it's actually a little bit opposite of what most scientific research does, but it's actually extremely powerful. So give us a chance to ask some question that wouldn't be dreamed of, but we can use this combinatorial selection to basically obtain that particular sequences.
How can DNA be used to control the shape of a nanomaterial?
The key word of DNA is programmable. And this is the so-called DNA hybridization. It's a double helix. This is discovered in the 1950s with the Watson-Crick basic base pairs. Not only fundamental how the gene is replicated, right? How our genetic information is stored and transferred into a next generation, but also this particular one is much more powerful than that. It's that one can actually programmable, so you can not only form a double helix, you can form triple helix, you can form not only linear, you can form two-dimensional, three-dimensionals. So basically, one of the really most exciting area of research in DNA nanotechnology is so-called DNA origami. So basically, just like a paper origami, any shape you can form, we can actually, based on that information, we can program the DNA sequences to can fold into the whatever shape you want using DNA. So that's why the DNA, because of the base pair ability, not only two strands of DNA, but multiple stranded DNA, they can form all kinds of shapes. Not only just cubes, they can form cylinders, they can form faces, maps or flags, or curved face surfaces, so corners and different kind of angles. So therefore, it's really exciting. The DNA really give you this nice shape controls that difficult to do by other methodologies.
How do you tune the selectivity of the functional DNA for different molecules?
If you have obtained the subset sequences, you can still fine-tune selectivity by understanding the fundamentally what each what is conserved as sequences, conserved residue that is responsible for the binding, and one can modify that and actually to achieve different selectivity. For example, one of the area of research we're interested in is actually tuning the so-called dynamic range for detection. So lead is a good example because for the lead detection in water, there's one threshold defined by EPA, so-called maximum contamination level. Below that threshold is considered safe to drink, above that is not. But on the other hand, same lead, but if in paint, it will be one threshold because people drink water, they don't eat paint. But even more importantly, even lead in the paint in the wall is different lead in paint in the toy because we want to protect the kid, because kids do associate with the toy more often than the walls. So therefore, having a lead sensor is not good enough. You have a lead sensor. On one hand, it can do tell them the threshold in water, threshold in toy, threshold in paint in wall, or threshold in the soil. So therefore, we need a vary that so-called dynamic range of detections. So that's one common modification come into place. So we have been able to use different modified sequences that we obtained from the selection. We actually modify so they can change the dynamic range. So one don't have to worry about we have one detector for paint, one detector for water, one detector for soil.
How is the functional DNA labeled?
So the way we do it, there's many different ways. I just give you a few examples. One is we're using the so-called fluorophore quenchers. So we put them spatially close. In one strand of DNA of fluorophore, another strand of DNA is a quencher. It's like a light bulb turning on and off. So in absent the target, we design a way that in the absent the target, the fluorophore will be quenched by the quencher, so have a minimal, if any, fluorescing signals. But in the present of the target, the binding of the target change the conformation of the DNA. You can actually release the fluorophore away from the quencher and light bulb turning on. So that's one nice way we can do it. In fact, I already have a commercial product based on this particular technology. Another way to do is using gold particles. Nanomaterial has been known to be extremely efficient in terms of signal transduction, depending on what nanoparticle you use. For gold particle, there's so-called surface plasma effect, whereas depending on the distance between the two gold particles, you can have different colors. When they're far away from each other, it's a very strong red colors. When they're close to each other, it's very strong blue colors. Utilizing that, we can use DNA to kind of a hybridization to bring the gold particle together, so they will be kind of a blue colors. But then in the present, the target, the function of the DNA can recognize the target, again conformational changes, and then you can actually make them distant far away from each other. You can immediately in second actually turn that blue color into red color due to the presence of the target, and then the selectivity coming from the functional nucleic acid.
So these materials can be used as MRI contrast agents?
We do the detection, fluorescent detection, mostly that in so-called in vitro tests, outside, using detection water or even blood or urine. But fluorescence is very difficult to penetrate through the skins. So one of the methodology we have developed is using MRI, because now I can do a three-dimensional non-invasive detection for metal ions or other small molecules or cancers or cancer biomarker, I should say. So then in this way, then you can actually do non-invasive detections. Why is it important? Because right now, in fact, I just took an MRI just a month ago for my own knees, because when you have MRI works beautifully when you have a tissue damage, or sometimes, unfortunately, for some patients have tumors. But that's actually too late for many people. So right now, the new state of art is that can we actually detect biomarker for these diseases, particularly, for example, cancer, before tumor develops. Therefore, you have to be able to have MRI contrast differences between the presence and absence of the biomarkers before tumor develops. So that's what we're actually developing right now.
Can you describe some of the work you've done to turn these into dipstick tests? How do those work?
Right. So what we have done before was that you can do the detector, but it's actually assays in the solution. So it works, but again, it's not very convenient because the user have to do a quantitative solution transfer. And for many people, you can do it, but for a lot of people, you can't, because it can introduce a lot of errors, can introduce a lot of uncertainty in the result. So one nice thing about Dipstick test is that we will develop, and many other people develop, but what we develop using functional nucleic acid based on our technology is that we can actually mobilize the DNA with the gold particles onto one part of a kind of a lateral flow device. So it's kind of similar to the pregnancy test, with one part of this piece of papers, and then in the absence of the target, and then when the blood or when urine or water flow through it, there will not be any conformational change. It will stay in one place. The color will be the kind of a blue colored. But then in the presence of the target binding to the functional DNA change the conformation, disrupt the binding, and go from the kind of blue color to aggregate into a small individual gold particles. So there's two colors. The two things happen. One's a color change from blue to red, but more importantly, because the nanoparticle is smaller now, it can migrate through the paper and can be captured on the other side of the piece of paper. Now I can have a separation, and then it can go from blue to red, but also the separation at different location. You can see the color change. So in this way, you can actually, in about something like less than a minute, you can see the color change. You can get information more like a pH paper or a pregnancy test, so you can get a color changes.
How are they used to target drug delivery?
We actually encapsulate the drug into a so-called liposomes, like lipid bilayers. We can capture a lot of these different drugs, and then on the surface of the liposome, we can put a functional nucleic acid on it. They can give a target. This will be that we select out in the laboratory the different nucleic acids that are capable of binding to biomarker for different cancers of different other diseases. So then this one, the drug will be safer, less toxic because it's being captured by lipid bilayers. But then when they actually reach to the target, in this case cancer cells, it can actually internalize it, and then it can dump their drug in there, so selectively binding to the cancer cell, not the normal healthy cells.
Can you describe your recent work to create coded messages within the DNA?
Right, so this is actually come out of the early discussion where I talked about reversible assembly. Because what we in that particular work, we're interested in how we can actually assemble nanomaterials in the reversible way. You can put in something in there. If you don't like it, you can actually take something out, replace it with it. So then we can think. So that's a very nice demonstration. But is any applications? One of the applications will be so-called encrypted messages in the spy game, right? So you can you send in somebody a piece of paper with no messages or something kind of random messages, and then you add invisible ink to it, and then the real message appears in there. So this is actually what we were able to demonstrate it. Then, but instead of doing that in the written messages, what we decided to do using the Morse code, right? So we actually the graduate student was able to do the embedded in the origami. It's a nanoscale kind of a spy game here now. So it's a nanometer size. It's a square messages. Then what we do that in there, we embedded the message of NaNO, nano, using Morse code. And then what we do then, we actually cover that with other type of material, so that when you received it, you will have no messages in there, just kind of random square piece of nanomaterial. But when you add a biotin to it, and then you can replace those places that have a weaker binders, and the one that remains are strong binders that coded for NaNO. So now actually when added, so basically biotin is an invisible ink, and then the message come out is a Morse code for NaNO. But of course you can encode other messages to your interest, but at a very nanometer scale. So you need a microscope to see that instead of just using eyes.
What other functions are you working on, and what do you think is possible?
So far our work has been mostly in vitro test. Dipstick technician, fluorescent test for environmental detections. So one of the major area of focus for us is right now to apply the same sensor, but make into more of a cellular detections, but more importantly, do that really in the in the human being or animal first, and then in the human beings. So it can really start telling people why the lead is so toxic, where does it go, where does the lead goes, what does it exert toxic effects, and if it's the iron is good, where does it function at different stages of development. Calcium is the same thing in a different stage of development, why calcium worked in different places. So we actually go much deeper into biological system. So far we've been outside the body, outside the cells. Now can we develop that inside the cells? So that's the first thing we are interested in doing. The second thing we're interested in doing is also fundamental understandings. Why this particular DNA sequence is so selective for lead. The other one, slight changes of different sequences. That one very selective for mercury. Another one very selective for calcium or magnesium. So we actually we're spending a lot of time to do fundamental understanding of it. So once we know more about the particular structure features responsible for these different metal ions, then we can actually allow to design even better or smaller, more stable, and more effective DNA sensors. So finally, and another area of research, we actually expand the scope of the detections, because there are always a new target coming through. For example, you're talking about flus, right? Every year there are different type of flus. There are different viruses. There are different type of new disease coming up. So can we actually, using this methodology, can very quickly select different DNA that targeting to different ones. Right now the process is still very long, and not necessarily always we get a target that's very, very effective. So a lot of trial and error process even for that particular method. Although we can always get the DNA that bind to the target, but there's no guarantee that can bind target very, very selectively or very, very tightly. So what we want to do then want to make this methodology even more effective, shorter time, tighter, and more effective. And for any new target, we can do maybe high throughput screening. And so the way we do that, again, we increase the throughput, but also we can introducing modified nucleic acid. So far, most of our work have been using regular nucleic acid, which is plenty already because of different combinations. Again, mentioned 100 trillion different possibilities, but still their functional group are limited. So right now we're interested in introducing different new functional group. Why not combining the amino acid, right, with the nucleic acid? So then can actually have a functionality of both. Now we actually have ability to bind to newer target better, tighter, and more selective.
What has your experience been as a Sigma Xi Distinguished Lecturer?
I'm really excited because this is really devoted to scientific research society. I think that to me, that's a fundamental to make our society better. I think through science. I think this is through science investigation, a lot of the whole society in terms of problems can be solved by using scientific research. A lot of political debate can be clarified by science facts. To me, I think this is really fundamental to everything we do in everyday life is a science base. So that's why I'm really excited to be associated. So this is my first time interacting with the society, but I'm definitely looking forward to more interactions.
What do you see as the role of Sigma Xi in the scientific enterprise?
I think there's a disconnect between the scientist and then citizens. And citizens kind of have a vague idea science is important, but they really don't know what we're doing, what the scientists are doing. I think that the Sigma Xi Society really play a key role in communications. I always dream about, we actually have talked to my colleagues, we watch night shows, we see all the actors and actresses, they got invited to talk about whatever they do. Sometimes actually pretty silly in my opinion, But on the other hand, I really dream about we can actually have a talk shows, like a night talk shows, talk about science on a regular basis. In other words, can we invite, Sigma Xi president or society members, but more importantly, a scientist, a Nobel Prize winner, to be a regular host on the night show, so they can talk about exciting work about that. I think if we can reach to that stage, I think our society will be so much better, because I think communication is the key, and then Sigma Xi play a key role in that particular one.